So I have to start by thanking the Hope Funds for this wonderful award. And it's really an incredible honor to join the distinguished colleagues who are tonight being honored and have been honored in the past. And also, I must admit, it's really wonderful to watch this sort of remarkable organization in operation. I feel extremely privileged to have been a part of the 20th century revolution in biology. I was only in junior high school when the 1953 paper announcing the double-stranded structure of DNA was published. And by the time I got to college, it was still too new to be in courses. I was fortunate to be assigned a work-study job in the lab of the late Alex Rich at MIT. And there I recall when I learned about the double-stranded structure of DNA, being completely enthralled because it offered the hope that there might be a molecular explanation to some of the genetic phenomena that had fascinated me when I was in high school. Yet, I decided that I should go to medical school because I'd simply never encountered a woman head of lab or a woman science professor, but I did know some women physicians. This all changed just the summer before I was about to go to medical school when I worked in the lab of the cell biologist Joseph Gall, who was then at the University of Minnesota. For the first time, he gave me my own project, and by August 1st, I was so exhilarated that I decided my future prospects didn't matter. I wanted to make scientific discoveries, and I was able to switch, as you've heard, to the graduate program in biochemistry and molecular biology at Harvard. So the new field of molecular biology that I then entered when I became a graduate student in Jim Watson's lab was very small, very eclectic. Everybody in the field was using simple organisms, bacteria or the viruses that infect them, to do their experiments with. And this was because it was reasoned that you could never learn anything about the underlying principles unless you started very simple. In fact, investigators who at that time were endeavoring to work on mammalian cells were sometimes even derided for taking on something that was so complicated we would never understand what was going on. So starting there, I think you can see that it was absolutely unimaginable to me what would happen in subsequent years. It was unimaginable that we would know the sequence of the 4 billion base pairs of the human genome. It was also unimaginable the impact this would have on medicine with new drugs and new diagnostics, and now even the prospect of precision medicine to treat many cancers and other diseases. I certainly never thought there would be a multi-billion-dollar biotech industry making products both for medicine, agriculture, and other things. The fact that forensic science would be revolutionized by DNA was unthinkable. And finally, it was inconceivable to me that women would ever hold important roles in science and in academia. I've also been surprised at how important serendipity is to the discovery process in science. In my case, it was chance comments by some other people that brought us a year or so later than that to using sera containing autoantibodies from patients with lupus, leading to the discovery of these little particles in cells called SNRPs. What SNRPs do is they're essential for pruning out the nonsense bits that interrupt virtually all of our genes and gluing the rest back together in a process called RNA splicing. So SNRPs, therefore, allow the information in our genes to actually get into the working proteins of the cells. So a final surprise for me has been finding that it's almost as much fun to share the joy of discovery with a younger colleague as it is to make the discovery yourself. And I've been just immensely fortunate to have a whole cadre of really fabulous younger scientists working with me at Yale, postdocs, graduate students, undergraduates. And it's to them that I really owe being here. And also the support of colleagues parents who encourage me to always take on a challenge, and the support of my husband and my son. And I just want to say thank you so much to the Hope Funds for doing the work that you're doing.